So just a reminder, this seminar series is co-sponsored by the P41 Funded Center for Mesoscale Mapping, housed in the Martinos Center. And today I have the privilege of announcing or hosting Dr. Christian Farrar, who is an associate professor of radiology at Harvard Medical School and an assistant in neuroscience at Mass General Hospital. He earned his BS in physics from the University of Wisconsin, Madison, and a PhD in chemistry from Harvard University. Following his graduate work, he did a postdoctoral fellowship at Francis Bitter Magnet Laboratory at the Massachusetts Institute of Technology. His current research focus is focused on development of novel MRI reporter genes and MR methods for molecular imaging. So please join me in giving a warm welcome to Dr. Farrar.
Well, thanks for the introduction. Great to be here. And what I'm going to talk about today is some of the work in my lab where we're trying to image oncolytic virotherapy. And why exactly oncolytic virotherapy? What exactly is it? So the idea is you take a virus, in our case, it's a herpes simplex virus, and it's engineered so that it can only selectively replicate in tumor cells. And so the viral replication eventually bursts the cell, leads to cell lysis and release of new viral particles that theoretically can spread throughout the tumor. So it's sort of ideal for treating maybe an invasive tumor. Not only that, it can also elicit a strong immune response against the viral infected tumor cells. So it's a form of immunotherapy. And you can even engineer the virus to express certain chemotherapeutics. So you can have a targeted drug delivery as well. So in theory, this all sounds great. In practice, of course, it didn't really work well in early clinical trials. Were found to be safe, but ineffective. So very little virus was actually found in tumor biopsies. And this failure was mainly attributed to the innate immune response. There's sort of a fine line between having too much of an immune response and too little of an immune response, and also to poor viral replication. And so there's been a lot of work ongoing trying to optimize the delivery and replication ability of the virus. Very recently, my collaborator, Nino Chioka, has published some work, clinical trial with a oncolytic herpes simplex virus engineered with a neurovirulence gene that led to preferential tumor replication and immunoactivation. So it's promising, but there's still work to do to try to optimize things. And that's where we wanted to develop tools so that we could look at detecting, first of all, the tissue molecular response to the virotherapy, but also ideally to be able to track the virus itself. Is it spreading in the tumor? Is it replicating? And in both cases, we've sort of been exploiting chemical exchange saturation transfer, CEST, MRI methods to do this. So we can assess the molecular response with CEST by quantifying the tissue pH and protein synthesis. As cells become apoptotic, the pH drops, protein production shuts down, for instance. And then the other aspect is we're also been working on designing MRI reporter genes based on CEST to allow us to image the viral spread and replication in the tumor. All right, so why CEST? Well, MRI, as most of you all know, is pretty insensitive. The only thing that saves you is that you're detecting water molecules and it's like 110 molar concentration. So you have very good sensitivity in the end, but you don't really have any molecular specificity. You're just looking at water, essentially. All right, well, magnetic resonance spectroscopy, on the other hand, has very good specificity for different molecules and metabolites, but you're looking at protein metabolite concentrations that are at best millimolar. And so the sensitivity is very poor. And so CEST tries to combine sort of the best of both worlds, where you have the specificity of the spectroscopy approach, but the sensitivity of water. So we're indirectly detecting proteins and metabolites through the water molecule, right? So we saturate signals on the protein that undergoes chemical exchange to the water. So it sort of acts as a saturation, a signal amplifier, if you will. So we have much higher sensitivity than spectroscopy. Now, we don't have quite the specificity of spectroscopy, but we still, you have certain characteristic signatures for different metabolites, for proteins, for lipids, et cetera. So it's sort of a compromise between the sensitivity of MRI and the specificity of MRS, sort of trying to get the best of both worlds. So how exactly does it work? So if we have a protein and we can saturate the amide protons on the backbone of the protein, and they can undergo, so we fill that signal, they can undergo chemical exchange with the bulk water. So we transfer that saturation to the bulk water. And if we have fast exchange, we can keep that saturation pulse on for a long time and keep pumping the saturated protons into the water pool so we get a big change in our water signal. Where again, fast exchange acts as this amplifier. All right, so conventional CEST, what do? We basically step our saturation pulse across the water line width. When we get on resonance with that exchangeable proton, you see a little shoulder there from that exchangeable proton. Of course, when you're directly on the water line, you get direct water saturation. So what's conventionally done is you subtract the left side from the right side of your so-called Z spectrum. Whoops, wrong way. To get this so-called magnetization transfer ratio asymmetry. And then you can see that the green is, you see a peak at the chemical shift of that exchangeable proton. Problem is, CEST contrast, it's just a weighted image contrast. It depends on a lot of different parameters, including the number of exchangeable protons or your concentration, the chemical exchange rate, which depends on the pH, your relaxation times, your RF saturation power and time, your saturation efficiency. So it's really only semi-quantitative. We're not really getting any of this information from just the simple magnetization transfer ratio asymmetry. It also takes a very long time to step all the way across the spectrum. You know, it can be 10 minutes or more. Not only that, when you're subtracting these two different sides of your Z spectrum, you basically are overlapping a bunch of different contrasts from different exchangeable protons. So on the right side, you have these aliphatic protons, which give rise to a relayed NOE effect. And if you subtract those from the amide side, then you have some complex overlay of different contrasts that can make it very difficult to figure out what's actually going on with the CEST contrast. So we really need faster, more specific and quantitative ways of extracting out some of this information. And that's where we kind of turn to magnetic resonance fingerprinting. So first proposed by Dan Ma and Mark Griswold's group at Case Western. And again, the idea is you have some noisy fingerprint, say at a crime scene. You scan that fingerprint for a match. And as long as the noise of the finger, you know, you can have a noisy fingerprint, but as long as the noise is not correlated with the pattern, the fingerprint pattern or your signal pattern, you can get away with very low resolution undersampled data. So, and you can get quantifiable information. So the MRI equivalent is we're going to vary our MRI acquisition parameters to generate unique signal patterns or trajectories for different tissue parameters. And then our database, we're going to simulate using the block equations. And then we're going to match those experimental patterns to our simulated dictionary. So for chemical exchange, the obvious parameter to vary would be saturation power, because if you have something with a very fast exchange rate, you're going to need to use high saturation power to efficiently saturate it. If it's slow exchange rate, you only need low power. So the logical thing to do first is to try just varying the saturation power. Here we varied it for 30 iterations. And then to generate our dictionary or our database, we have chemical exchange between the water pool and our solute proton pool. And we're going to use this acquisition schedule and then use the block equations and just simulate all possible combinations of relaxation times, proton volume fractions, and exchange rates. And that's the signal you get for that particular combination. That's one element of our dictionary. And we just plug in all these different possible combinations of parameters to build up our database or dictionary. So then to do the matching, here I'm just showing a simple example. This is L-arginine, where we're going to saturate the N-alpha amine protons, exchangeable protons, varying the saturation power for 30 iterations. Again, we don't care about being in the, we don't want to be in the steady state, so we can use a short repetition time. So this was 2-minute acquisition. And then we're going to match it to our simulated database. So this is just showing you an example of that process. We take the 2-norm normalized signal pattern or trajectory. In red is the experimental signal pattern trajectory, and in blue is an example of a simulated pattern for an exchange rate of 400 per second and 25 millimolar for L-arginine. And you see you don't get a good match. So a perfect match would be a dot product of 1. And then you can try other possible combinations, 100 per second and 10 millimolar, still even worse. But here for about 1200 per second exchange rate and 57 millimolar, you get a very good match. Now it's not perfect. And part of the problem is, you know, when you have a lot of parameters that you're simulating, your dictionary gets very computationally difficult, untractable. So frequently you won't have very high resolution in your parameters you're simulating, and that's going to limit your accuracy. And I'll return to that in a little bit. But so here's just showing that example of that L-arginine phantom. Here are 2 different phantoms, one where we varied the concentration and one where we varied the pH. And you can see the top one, the pH is fixed, the concentration is varied. And so since the pH is fixed, the exchange rate should be fixed. And you can see indeed it is. And then the concentrations, we get very accurate concentrations for the L-arginine. And then conversely here, where we vary the pH, you can see that the exchange rates varying, whereas the concentrations are fixed. And so with the fingerprinting, we're actually able to separate out these 2 contributions, the exchange rate and the concentration, and quantify them separately. Now, of course, once you go in vivo, things become a little more complicated because you don't just have, say, an amide proton. You also have this somewhat annoying semi-solid macromolecular pool of exchangeable protons that's very broad. So here in green, that's the MT pool. And when we saturate, say, on that amide proton, we're also going to be hitting that MT pool. So now we have to simulate an MT pool as well as our amide pool. And again, you know, your dictionary kind of explodes exponentially with the number of parameters you're trying to match. So it becomes very challenging, and your accuracy, again, you're going to be limited to the resolution. You can simulate all these different parameters, and that's going to affect your accuracy. And so we moved to sort of implementing a deep learning neural network to try to do this reconstruction of our parameter maps instead. And so we first try and simplify and isolate the semi-solid and the amide pools using sequential targeted fingerprinting acquisitions. So we vary the saturation power, and then we vary the saturation frequency offset here between 6 and 14 ppm. So we're just hitting the semi-solid pool. And we input those images along with T1, T2, and B0 maps into a neural network to generate these semi-solid proton volume fraction and exchange rate maps. And then we can plug that into another neural network where now we're saturating at the amide proton chemical shift offset, but we're giving it the information from so we've already pinned down the parameters for the MT, the semi-solid pool.
The exchange rates on those macro exchange rates?
They're pretty slow. It's, yeah, on the order of like 10 per second. I mean, one of the problems with CEST is that no one really knows. I mean, because there's no ground truth gold standard for measuring these things. So, but generally it's pretty much agreed that the
You can't really go way off resonance because I mean the macromolecular pool.
It's very broad.
Water, right.
Yeah, I mean, I'll get to later. We have worked on trying to optimize these acquisition schedules, and yes, this schedule is not the one that is optimal, probably. So anyway, this allows us to sort of isolate the 2 different pools and get them and pin down one, and then use that information to get the other for the amide proton. And the nice thing is we can train the neural network with a sparse dictionary, so it's not so computationally expensive. The neural network outputs a continuous range of parameters. You don't have this problem of just, simulating discrete values. And then also it's much faster. So if you have some huge dictionary you've simulated, it can take many hours to do the dot product match with every element of the dictionary to try to find the best fit. Whereas the neural network, once it's trained, it's all less than a second to get these maps generated. All right, so finally getting back to oncolytic virotherapy. So here what we did was we have a mouse U87 brain tumor model imaged at baseline and then 48 and 72 hours after injecting the virus into the tumor. And so we have T1, T2, water T1, T2, the semi-solid exchange rate, proton volume fraction, and then the amide exchange rate and proton volume fraction. And what you can see in the amide exchange rate, is there a pointer? Anyway, the tumor here, you see the exchange rate is slightly elevated at baseline. And also at the later time points in regions where the virus is not, in non-viral infected tumor regions, the exchange rate is elevated. And this makes sense because we're looking at sort of the small mobile proteins that are primarily in the intracellular space. And it's well known that the intracellular pH in tumors is actually slightly elevated, whereas the extracellular pH is decreased. So you get this inverted pH gradient in tumors, but the intracellular pH is actually increased. And we see that. We see an increased exchange rate in the tumor. However, after injecting the virus, you can see in the center of the tumor where we injected the virus, at 48 hours, there's a small region where the exchange rate is decreased and also the protein concentration. And then at 72 hours, you can see that area of decreased exchange rate and protein concentration that has grown bigger. So the virus is replicating and spreading through the tumor. So you can actually see that reflected in the pH and the protein concentration where they're decreasing. And again, this is consistent with apoptosis because it's well known that at early stages of apoptosis, you get a dramatic decrease in the pH and also protein synthesis starts to shut down. Okay, and then in the semi-solid proton volume fraction, it's also decreased, which kind of makes sense because you're replacing sort of highly myelinated brain tissue with tumor tissue. And well, how do we validate this? So we did some histology looking at apoptosis biomarkers using caspase-3. And we see that the viral infected tumor core is indeed caspase-3 positive. Similarly, we can stain for protein content using this Kumasi stain. And we see decreased protein content in that same caspase-3 positive tumor core. And we get pretty good localization of the histology regions with the regions of decreased exchange rate and proton volume fraction. Okay, so just for all the mice, you can see again, you get a significant increase in the exchange rate or pH at baseline, but a significant decrease in the apoptotic tumor regions, again, consistent with decreased tumor cytosol pH. And then the proton volume fraction at baseline is actually decreased with respect to contralateral tissue. This is probably maybe due to tumor edema, decreasing the protein concentrations. But then you see a very significant decrease in the apoptotic tumor regions compared to the more normal tumor regions. All right, and then just for comparison, this is conventional CEST, this MTR asymmetry. And you can see the contrast is elevated in the tumor, but there's no significant difference between the apoptotic and normal tumor regions. And, traditionally people always argued, oh, the increased CEST contrast, amide proton contrast is due to increased protein content. But at least we certainly don't see that. And I think a lot of what people are seeing, you have this complex overlay of the NOE, the aliphatic protons, on top of the amide. You also have edema, and so your T1s are longer. So then, of course, your CEST contrast is also higher. So I think a lot of what people used to argue about increased protein concentrations is actually kind of not true in many cases.
It's pretty well.
And then you get longer T1, so your CEST contrast is higher. So that's why I think it's really useful to be able to, pull out these different contributions to the CEST contrast, because there's actually a lot of useful information buried underneath all those different contrast mechanisms. And just also to point out, there are no differences in T1 and T2 or diffusion ADC between the apoptotic and non-apoptotic tumor tissue. So again, I think using this quantitative CEST MRF approach, we can provide much more early insight into the tumor molecular response to the virotherapy.
The ADC doesn't go up?
Well, it does go up in the tumor, but I'm saying there's no difference between the apoptotic tumor region and the non-apoptotic tumor region at these early time points. So now we're working on trying to translate this to the clinic. So here's just showing a normal brain on the Siemens 3T Prisma. This is using a 3D single-shot EPI with 1.8 millimeter isotropic resolution. And again, it's not too bad. So if we acquire both the MT and the CEST schedules, that was about 5.5 minutes. Now we also have to acquire a T1 and a T2 map and a B0 and a B1 map. So all told, you're talking about 11 minutes. That's kind of on the upper end of what someone's going to tolerate for clinical scans. So we really need to be able to do better and bring these acquisition times down, ideally. So that's where we've been trying to work on how to optimize these acquisition schedules so that we acquire as few images as possible. Well, Nina Choka is doing a clinical study. The problem in the past was, this was before, say, Daniel was over there, is that, our sequence is sort of a pulse-seq hybrid. So it's a hybrid sequence. So the pulse-seq is used for the saturation, and then we have a normal Siemens readout. And they wanted nothing to do with that over at the Brigham previously. Pulse-seq, that is. That's very different now. And actually, Siemens actually has a whip now that incorporates pulse-seq for the saturation aspect. But back then, no, it was really hard to get them to put my sequence on over there. So yes, yeah, now the clinical trial is paused. I think they're going to start it again. So, Daniel and Florian over at the Brigham are starting to work on getting these sequences, the new Siemens whips. There are lots of issues. Most of them have to do more with the 7T scanner, but they're also getting them on the 3T scanner. So hopefully that we will be able to do that there soon. But again, it's still kind of long, 10 minutes. So can we cut that down? And when you start cutting down the number of images you acquire, it does degrade your ability to discriminate your different parameters. So how do we optimize our acquisition schedule? Oops, wrong button. Right, so how can we minimize the number of images we need to acquire to determine, is that a dog or an ice cream cone or a dog or a mop, right? And if we can increase our ability to discriminate, we can take fewer pictures and allows us to reduce the scan time. Problem is, of course, optimization of your acquisition schedule is very challenging. You have a huge parameter space, both your number of acquisition parameters you can play around with, but also your tissue parameters. So how do we go about doing this? And that's where, in collaboration with Matt Rosen's group, with Bose Zhao and Matt Rosen's group, they had developed this thing they called AutoSeq for basically you have a computational graph form of the block equations to try to optimize your pulse sequence. And so we just slapped on our saturation pulse in front of that. And then we also added the neural network on the other end. So we have sort of a joint optimization at each training step of both the acquisition parameters and the reconstruction neural network. And this was all done by Or Perlman, again, in collaboration with Bo. So here just to show a very simple example. We have a range of chemical exchange rates. Endogenously, the amide protons are around 30 per second, and a range of volume fractions. And we start out and we want to simulate different signal trajectories for these different combinations of parameters, and we want to make them as distinct as possible. So initially you start out with no saturation power, just have a short TR, which is why it goes down after that first shot. But now this auto-CEST neural network is trying to optimize. So each one of these traces is a different combination of chemical exchange parameters. And so it's trying to optimize the difference between these different signal patterns by varying the acquisition schedule. So this is just a very trivial sort of example, just to give you an idea of what it's doing. And then here's another simple example. This is iohexol, which is a CT contrast agent. It has two equivalent amide protons. And what we did is we started with that L-arginine acquisition schedule we'd used originally, which was 30 acquisitions, and we just truncated it to 10. So we're only acquiring 10 images. And if you just look at that L-arginine schedule and you do your fingerprinting with that schedule, it's garbage, right? Everything's just pegged to the maximum value we had in the dictionary of 100 millimolars. So this again points out that just using any random schedule, especially when you're making your schedules very short, they're not all created equal. Some of them will give you garbage. So it is very important to optimize it for the case of interest. And then you can see also, the exchange rates here where the pH is constant, here they're varying. So again, it's not doing well on the concentration or the exchange rate. But the auto-CEST optimized schedule, then we're getting very good concentrations and exchange rates. So now we're starting to apply that to optimize both an amide schedule and an MT schedule for the brain. We got some preliminary data in my brain. And it didn't really change much, but because I think the schedules we started out with for the amide and for the MT were not that bad. But for other cases, it may be more important. And again, yes, we do want to try to start imaging some of these patients that are undergoing oncolytic virotherapy over at the Brigham. And then we also recently got a grant, Daniel and myself, to extend, CEST fingerprinting for the quantification of glutamate in GBM patients that are being treated with therapies that affect glutamate metabolism. All right, so that's sort of the first part of the talk that was focused on, let's look at endogenous changes and molecular signals in response to the virotherapy. But then the second part is, can we actually image the virus itself with the reporter gene? So, again, we'd like to be able to see what's going on when you inject these new viruses that are modified. Are they replicating? Are they spreading or not? And it would be great to have something that's expressed endogenously. There are some PET radiotracers, thymidine kinase, for instance, but you got to get your radiotracer there, and it has to be retained in the cell. And once you get a lot of cell death, then your PET radiotracer signal decreases, and even though you have a lot of virus there. So it would be nice to have something that's expressed endogenously by the tumor cells. So is there sort of an MRI equivalent of a fluorescent reporter gene? And that's where we, again, turn to CEST using chemical exchange to try to detect some protein reporter. So originally, this was a collaboration with Saf Gilad, who was at the time at Johns Hopkins. And he had developed this lysine-rich protein, LRP reporter gene, which consisted of 200 lysines. Why lysines? Well, it turns out if you have all lysines and the amide proton exchange rate is quite fast, it's around 400 per second. Whereas most endogenous proteins, the exchange rate for the amide protons has been measured to be on the order of 30 per second. So anyway, he cloned that into a tumor, 9L glioma cell, implanted into mouse brain, and you can see the LRP expressing tumor. You see elevated CEST contrast compared to the control. So we took that LRP and put it into our oncolytic virus. This was G47 delta oncolytic virus. And it does work. So you can see the LRP expressing viruses. You see elevated contrast after the virus injection compared to controls, but it's pretty small. You know, you're talking about a 1% change. And you have the issue of specificity because we were looking at very early time points where there's not a lot of change in the background CEST contrast. But at later time points, as showed in the first part of the talk, you're going to get all these changes in the background CEST contrast due to the response to the therapy. You know, the pH is going to be changing, the concentrations are going to be changing. So how do you separate out the background signal changes from your reporter protein signal changes? So you have a specificity, wrong way. Not only that, because it's a very repetitive sequence, the DNA sequence for it, because it's all lysines. What happens when you have very repetitive sequences is that you end up getting these things splicing in and out, the repeats of these lysines. And so instead of getting full-length protein, you get sort of here in the Western blot, you see a smear between, I don't know, 10 and 20 kilodaltons, whereas full-length protein should be 36 kilodaltons. And if you look at a southern blot of the plasmid DNA, you don't see a single band, you see a whole ladder of bands. All right, so that's another problem with the sensitivity, that we're not really expressing a stable protein. And again, I mentioned the specificity, we have these background changes. All right, so we really need improved reporters and methods with increased sensitivity and specificity. So the first thing we tried to do is, all right, let's try and make it stable, break up these lysines with other amino acids. So we screened a library of peptides where we have certain amino acid motifs to break up the lysines. And we just took the ones that perform best and we linked them all together in a protein. New reporter protein. It's smaller, it's only 100 amino acids and with 70% lysine content. But it has no DNA repeats, no GC-rich regions. It's much more stable protein. It's less highly charged. These should all make it much better as a reporter. And you can see then here that we get full-length RNA expressed, just a single band observed. We don't get all these truncated fragments. So this is just the new reporter into a cell line. And then we took cell lysates. In red, you can see, because we now have arginine, you get these guanidinium protons that resonate around 2 ppm. And then these are the amide protons. And so it's increased in the cell lysates. And very similar to if you look at the peptides, this RHGP peptide with the arginine, you can see that same peak at around 2 ppm. And so then we looked at cell lines engineered with this reporter, and you do slightly better. You have about 2.5% CEST contrast, so it's comparable to what we observed with the first generation sort of LRP reporter. But again, it's still pretty pukey, pretty small. And so we really want to try to optimize the sensitivity. Well, how do we do that? So one approach we took was to try to use essentially machine learning. So natural evolution has only explored sort of a small region of the protein design space. And most people frequently use directed evolution to search for better proteins, say through mutagenesis, but this is very time consuming and a lot of the mutations are dead ends and don't do anything. And basically, you know, it's the problem of if this is your global maximum, your best protein, and you start here, okay, maybe you'll find it. But if you start here, then you can get trapped in some local maxima, and it can be difficult to find that one. So the approach was to use a genetic programming approach where you sort of sample the whole protein design space, and then you rely on convergent evolution of proteins with different origins but similar features, where after many evolutions, you start converging on that global maximum. And how does this work? Well, basically, we started with like 100 different models, and each model consisted of 50 different amino acid motifs, which each have a certain weight in terms of how good they are for the CEST contrast. And then you look at how these models, what CEST contrast they predict, and then you compare it to the actual experimental data you get for certain peptide sequences. And then you evolve this over many generations. You take the best 40 models, and then you can also have recombination, random mutation mechanisms where you say mix up some of the amino acid motifs or change some of the weights in order to try to train this genetic programming tool to predict what is going to be the best proteins. And so after, about 10 generations, we were finding peptides with 4-fold higher CEST contrast compared to polyol-lysine. And it wasn't entirely due to the faster exchange rate, but we were getting peptides that had 2 times the exchange rate of, say, the polyol-lysine amide protons. But some of it was also coming from getting additional things like the guanidinium protons contributing to the contrast. But at the end of the day, to make a long story short, we're kind of bumping up against the limit of what we can do with these amide protons. Limit specificity, again, endogenous proteins have the same chemical shift. By optimizing the exchange rates, we can gain some specificity and selectively try to, excite these fast exchanging ones. But the problem is, eventually, because of the small chemical shift, amide protons, 3.5 ppm from water, CEST, ideally, we want to be in the slow exchange regime where the exchange rate is less than the chemical shift difference. And so we hit some ceiling where now our chemical shift is too small, so we can't access these higher exchange rates because for these faster exchanging reporters, the amide protons are difficult to saturate efficiently without inducing significant direct water saturation. Not to mention that high saturation power has become difficult to implement on clinical scanners. So one possible solution is to come up with reporters that have much higher chemical shift so we can access these faster exchange rates. So if you wanted a protein that's expressed endogenously, we're limited. There aren't many candidates, but basically what has large chemical shifts? Well, a lot of aromatic ring protons. So histidine, imidazole, NH ring protons, the tyrosine, phenyl, OH, tryptophan, indole, NH protons. All can have shifts, anywhere from 5 to 13 ppm from water. One of the challenges, though, that these exchange rates tend to be extremely fast, sometimes, 10,000 per second or more, so that they aren't frequently observed. And again, it's very difficult to saturate these things that are very fast. Not to mention that aromatic groups generally we think are hydrophobic, so how is the chemical exchange with water going to work? And so, typically you don't really see any CEST signals at these higher chemical shift offsets from most peptides and proteins. So there are some examples. Here, this is superoxide dismutase, which has 6 histidines coordinating copper and zinc ions, and you do get CEST contrast at 7.3 ppm. And the exchange rate is fairly fast, about 800 per second. The problem is this is going to be hard to engineer a reporter that has maybe multiple repeats of this sort of structure. So another thing we looked at was tyrosine-based reporter. This is basic pancreatic trypsin inhibitor, BPTI. The problem is these tyrosine, the phenyl OH protons exchange extremely rapidly. So here you can see at 4.8. When we're at low pH and low temperature, we can see these peaks. As we increase the temperature and the pH, though, they basically broaden out and we can't really detect them because the exchange rate is just too darn fast. So then tryptophan, another possibility. Here we kind of stumble across this. When we were searching for amide protons with fast exchange rates, we suddenly, in one of the peptides that had a bunch of tryptophans, we saw this peak at 5.4 ppm due to the indole NH proton on the tryptophan. And it had a fast exchange rate about 740 per second. So this made us think, well, okay, how about using tryptophan-based reporters? But we had looked at other peptides that had tryptophan, and they didn't show any peaks. So our question was, well, why does this particular peptide show some peaks, some signal from the tryptophan? And so we started trying to play around with the amino acid motif to explore the importance for the contrast at this 5.4. And one of the things we did is we removed the glutamate in this sequence. And much to our surprise, suddenly we saw this huge peak now at 4.2 ppm. That's kind of almost swamping the amide peak with an exchange rate of 1900. And we had no idea what this proton was. So Dave did some peptide structure predictions. And what you see is the lysine, the side chain amine, is in close proximity to the indole ring of the tryptophan. So we're getting this sort of cation pi interaction. So what we thought is, well, maybe this interaction is shifting the amine proton on the lysine, which usually is at 3 ppm from water, shifting it further downfield, and also slowing down the exchange rate, because usually you don't see these amines because the exchange rates are too fast. Okay, so in order to investigate this, we started substituting things for the lysine. So ornithine is equivalent to lysine, except the side chain is 1 carbon shorter. So if you substitute ornithine for the lysine, the signal disappears, that 4.2 ppm signal. Similarly, if you substitute 5-hydroxytryptophan for tryptophan, at the 3 position, it disappears. At the 10 position, it's a little more ambiguous. It does have an effect, but it's not quite as dramatic. So it does indeed appear that we have an interaction between the indole rings with this cationic amine group on the lysine side chain that's shifting the frequency, the amine frequency downfield, and also leading to a decreased exchange rate that's now more favorable for CEST. So then this got us thinking, oh, are there other proteins out there that have cation pi networks? And it turns out way back in 1999, Dennis Doherty at Caltech had searched the protein data bank looking for proteins with cation pi networks, and found one really striking example, glucoamylase, where you have this lysine, the side chain amine, is sandwiched between 2 tryptophans and 2 tyrosines. So it's conveniently available from Sigma, and we tried it out, and sure enough, you get a peak at 9.7 due to tyrosine. And again, it's very broad because it's still kind of too fast, but at least you can see it at physiological temperature and pH. And then here's the tryptophan at 7.3 ppm. And then there's kind of this unresolved peak at about 4.4, which we think is probably the lysine, analogous to the peptide, the amine proton. And the exchange rate for the tryptophan was 2,500, but it's still in the slow to intermediate regime because the chemical shift is so big. So it's still favorable for CEST. The tyrosine, again, that exchange rate's still too fast. And in this paper, they found that over 25% of tryptophans are involved in cation pi interactions, and that these cation pi networks are found primarily on protein surfaces. So it gives us hope that they'd be good for CEST and exchange with the bulk water pool. So yeah, so here's just an example comparing glucoamylase versus polyl-lysine. First, we do the amide contrast, and again, glucoamylase also has amide proton, so it gives contrast. It's not as high as the polyl-lysine because the polyl-lysine, the exchange rates are much faster. So you get contrast from both, but if we look at 7.3 ppm, the CEST contrast, the large chemical shift of this indole NH provides specificity. So you don't see anything from the PLL at the different saturation powers, but you do from the glucoamylase. Now, of course, one of the issues is that because the exchange rate is so fast at low saturation powers, you don't see much, you see a little bit, but you really have to go to higher saturation powers to detect a significant signal. But this fast exchange rate is providing a sensitivity because, again, from mutation studies, the 7.3 ppm signal from the tryptophan was assigned to one particular tryptophan. So we're basically comparing one exchangeable proton versus for polyl-lysine, it had about 65 amide protons. So if you compare it on a per exchangeable proton basis, at 2 microtesla, you get about a 5-fold increased sensitivity relative to polyl-lysine. At 5 microtesla, it's 17-fold, and at 9 microtesla, you have 37-fold. Again, just because the exchange rate is so much faster, you get a lot more bang for your buck from the glucoamylase versus one of the amide protons. But high saturation powers are still an issue, especially for clinical translation. So this is where we then start thinking, we saw in the literature some work from Moritz Zeiss's group, where they were looking at chemical exchange spin lock. And basically CEST can be thought of as a spin lock experiment. You're just spin locking off the water resonance, you're spin locking on your exchangeable proton. And if you look at the equations for the effect on the water the chemical exchange induced effect on the water relaxation, there are kind of two terms. You have this water resonance spin lock term, where delta omega is the offset from water. And you also have a CEST term, delta omega B, the offset from your solute chemical shift. And in the slow exchange regime, so exchange rate of about 100 per second, the contributions from the on-resonance spin lock versus off-resonance, they're about the same. So there's no good reason to do the on-resonance because there you see all exchangeable protons. But if you go to higher exchange rates, the intermediate exchange regime, so here it's about 1,000 per second, you get a much bigger contribution to the exchange, the water exchange, the water relaxation from exchange from the spin lock versus the off-resonance saturation. Now, again, you lose some specificity on the on-resonance spin lock because you're going to see all exchangeable protons, but they are going to be weighted to those with large chemical shift. Delta omega B is the chemical shift in water, and that goes squared, and fast exchange rates. So hopefully we can get away with some of this. And here again, just demonstrating glucoamylase and polyl lysine. This is the CEST spectrum, and this is the spin lock. And you can see you get very large contrast for glucoamylase, basically almost nothing for the polyl lysine. So you get much higher contrast using the spin lock for these fast exchanging protons. And you also notice that as a function of saturation power, 2 microtesla 5, 9, there's no difference in the contrast for the spin lock approach. And that kind of makes sense because if we're on resonance, delta omega squared is 0, and then these omega 1 squared terms, the B1 saturation power drops out essentially. So it's almost essentially independent of the B1 saturation power to use the spin lock approach. What?
You have tubes in the bottom, the spin.
Yep.
You know, you have great contrast, right, for the glucoamylase, but then you see contrast in the tubes that are for the polylysine. It's not the polylysine.
Not much.
Well, no, but you have that ring. What is that ring?
That's that's just a glass tube.
So that's just no signal. That's just no signal. Okay.
Yeah, no signal.
Yeah. Contrast.
Yeah.
Right. No, I'm just saying because it looks the same, and you don't see the tube in the upper one.
And again, you know, here we had to use long saturation times, 10 second. Here we have a 300 millisecond spin lock. So now and again, this contrast is going to be dominated by things with fast exchange rate and large chemical shift. So for instance, the amide and aliphatic protons, they have slow exchange and small proton volume fractions. So they're way down here, simulation. But the hydroxyl protons have fast exchange, but they tend to have small chemical shift and proton volume fractions, so they don't contribute too much either. So the main contribution is from amine protons, say, or semi-solid MT proton pools. The semi-solid, they have very slow exchange, but they have humongous concentration. So it's a bit of a problem, but we can use the same approach where we use a sequential. We lock down the MT parameters first and then feed it into those parameters into the next neural network. So it's mainly the amine protons have fast exchange, and the semi-solid protons have slow exchange, but very large proton volume fraction. But in order to try to get some specificity, what we decided to try first was sort of mixing and a fingerprinting acquisition that had both spin lock and CEST components. So the spin lock provides high sensitivity, the CEST provides high specificity, and we combined them together in one acquisition schedule. And originally, we were doing this for our glutamate project, but it'll work equally well for our reporter gene project. So, but glutamate, amine protons at room temperature at least have an exchange rate that's basically exactly the same as the glucoamylase, so 2,500 per second. And so using this hybrid spin lock CEST fingerprinting method, we quantify the concentration exchange rates, and you can see we're getting on the right is the error map. We're getting very good concentration values using this sort of approach for glutamate. And glucoamylase has a much larger chemical shift, so it should be even more sensitive for the glucoamylase, because again, it goes like the square of the chemical shift. Or if we compare it to polyl-lysine, we not only have a faster exchange rate, but we also have a much bigger chemical shift. So we should get much better sensitivity. And so we're now working on trying to do this sort of hybrid approach for the glucoamylase as well. So then finally, again, we're starting to screen peptides and proteins with cation pi networks. We want to try to isolate some small optimized structure and then engineer multiple repeats into a reporter protein. And then finally, again, optimize this spin lock CEST method to try to quantify things. And then just finally close by thanking all the people who've been involved with this project over the years and NIH funding, in particular Dave and Chetan right now, and formerly Or and Ori and Jonah and Shuning, who all were involved in this project. And thank you for your attention.